The processing practices associated with the slaughter of cattle have long been improved to enhance both the animal's till-death welfare and the food safety standards for consumption. One of the key concerns has been Escherichia coli or E. coli contamination of the meat. This is attributed to the fluids from the digestive system of the cattle contaminating the meat whilst undergoing post-slaughter practices. Ongoing research is now showing a correlation between grain feeding of cattle and increased E. coli levels in their digestive system. The E. coli breaks down the starch in the grain, and therefore levels are increased with higher proportions of grain in the diets. The scientific journal article, Grain Feeding and the Dissemination of E. coli resistant bacteria from cattle, investigates the role of E. coli in the digestive system and its proportional levels associated with nutrition received by the livestock. The aims of the study were to explain the increased incidence of foodborne illness, to improve food safety and continue to maintain high industry food safety standards for meat production, to investigate suspected more rapid evolution of bacterial virulence factors, to maintain and improve the food hygiene standards and implement strategies to best overcome the impacts of the bacterium, to investigate the role of acid resistance in the dissemination of pathogenic bacteria, to determine the correlation between the type of feed cattle were fed and number of bacterium in their digestive tract. To determine the impact of these aims, the following method was used. Colonic digesta were removed from the rectum of cattle that had been fed hay, grass and varying amounts of rolled corn. The pH of the digester was tested by a combination electrode followed by a measure of turbidity to determine the level of coliforms. The bacterium were also tested for acid shock with lauryl sulfate broth to determine the alkalinity of the colonic bacterium and their ability to survive a change of diet. For example, the study was conducted with a herd of mature, non-lactating Holstein cows that were fed every 2 hours on diets of 0%, 45% and 90% grain. The results of the experiment demonstrated that grain supplementation had a small effect on the numbers of anaerobic bacteria in the rumen, but the colon there was a 1,000-fold increase. Strains of bacteria were very similar between each feeding regime, but the grain-fed bacteria had more acid shock survival. These findings could potentially have massive impacts on the food safety, as this could lead to greater harbouring of pathogenic strains and potentially increased dissemination of E. coli from cattle to human. Grain feeding has long been used to improve productivity and efficiency of cattle industry. This report demonstrates the impact of grain feeding on the virulence and numbers of E. coli in the digestive system of cattle. The recommendation of the author was a brief period on hay before slaughter to reduce the risk of foodborne E. coli.